Welcoming everyone. This is a great and more pizza. So with the pizza and science meeting. Secondly, I would like to thank Suhail, who's not here, for really doing the lion's share of the organization and his staff, Michelle and Tasneem. It's been really extremely well organized. So today, it's really an honor to have Mark Nicolls be our speaker. Many of you know Mark, so I'll be very brief. Mark is currently the division chief of Pulmonary, Allergy, and Critical Care Medicine, and he is the associate dean for research in the School of Medicine. And Mark actually is our fearless leader here at Arweh Hall. His meetings are excellent. He's always trying to advocate for us everywhere in the school and at the level of the university. So thank you for doing that, Mark. Let me just, on a personal note, say that the best way to know a person is by knowing who knows them. So, even though Mike and I had never met in person before coming to Stanford, two individuals told me about Mark. One is Bruce Paul, who used to be an immunologist and a nephrologist like myself. He used to be at Stanford many years ago before going back to Melbourne, Australia. And Bruce can be credited really with the discovery of CD4 positive regulatory T cells, seminal work done in transplantation. Of course, he was overlooked for the Nobel Prize last year. And when I told Bruce that I'm going to Stanford, he said, you should go and meet Mark. So that's Mark, honor number one. Number two is a person by the name of Mike Baldwin, who's currently the dean of the medical school at the University of Maryland. And again, I told Mark I was moving to Stanford, and he said, there's a guy there called Mark, and he's the one I go to for advice. So Mark, I'm really pleased to be a colleague of yours. Welcome to the Pizza and Science Lecture.
Oh, thank you very much. Thanks, everybody. Oh, yeah, so everybody saw the upcoming stars that are going to be talking.
Oh, yes. Do they have a slide for the upcoming lecture?
Yeah, do you want to?
Yes, very sorry. Apologize. I always forget to admit. So as you can see, we have speakers scheduled till October 1st. So I think that's a great sign, and we hope to continue this next year. So please sign up. Could be either a faculty member or a member of the lab here who could speak on behalf of the lab.
Well, while we're transitioning through, I want to thank Fadi and Suhail. Fadi actually was a hero of mine. He didn't know me, but I knew him. He's been famous in transplant for many, many years. And we were so honored that he came back to us. I also want to thank everybody for coming today, because I think this is what we need. This is exactly what we were just talking about at our building meeting, about how SRB might be a better place, would be a better place to be if more people were physically here rubbing elbows. We know that it's also hard. People in an expensive area, Joy and I deal with this all the time with, how do you live in the Beverly Hills of Silicon Valley and, have people drive into work, and the whole geopolitics of this place is complicated, and we know that it's not easy to be here. But when you are here, this is what it used to be like in the old days, and we miss the interaction of cross-disciplinary stuff. So we're still waiting. What else can I say? Okay, well, what I'm going to be talking about today, by the way, I made this talk when I thought it was an hour, and then I heard later it's supposed to be 20 minutes. Okay, I'm going to be light and brief, and I already had made the full talk. And basically what it is like, it was an exercise in trying to use AI to figure out how in the heck did we do, why did we do what we did, for the last, since 2007. When I first came so I did medical school here and residency, and then I went to Colorado for 11 years, and I came back, and really we've decided. We've decided to. We started off with this interest in the microvasculature. So what I'm gonna, oh, and what I want to do also, and you may have to shout because I can't see all you, just stop me, because this is supposed to be a friendly, interactive meeting. And because I have too many slides, which I'm gonna go through really light, I'm not gonna go deep into the science, I just want to give the big ideas. I would really welcome people to interrupt me, and we can talk about any particular thing that you want to as we go through this, and keep it interactive. And I promise I will be done on time. So you will not leave this room late. So what I want to do is talk about our group's research here, and what does it mean? What's the theme that's been uniting our research? And it's really this vascular immunobiology concept, and the concept that I'm going to be developing through this talk. Oh, and I also want to use it as an announcement for Dr. Ying Zhang, who's a bioengineer who's going to be on this floor, wet lab, who's joining our division. And she is works with in silico microvasculature research, and it'll dovetail with many people in the room that are interested. And she's a wonderful and collaborative person. So why don't I get started then? So first I want to thank all the key contributors. You're going to recognize Amy and Jingwo probably on this slide, maybe Dongyong and Tim and others. I'm going to be going through, but I really want to acknowledge their very hard work in the science that I'm about to present, and I'll be naming them along the way too. So let me start with lung transplantation. This is something that I do in clinic. I take care of patients after they get a lung transplant, and it has the worst survival of any transplanted organ, and only about 50% make it to 5 years survival. And so we were really interested why this happened. So when I was still in Colorado, I developed an orthotopic tracheal transplant model. There was one other tracheal transplant model out there that was orthotopic, meaning that we sewed it in place, and we still do this to understand how the airways work. Turns out the airways are really good for studying the microvasculature. We became interested in the microvasculature is because I was giving a talk like this, and one of the scientists in the crowd at Colorado said, "Why are the blood vessels so weird?" And we knew that this guy named Donald McDonald at UCSF was studying the microvasculature, and we learned how to use his technique and apply it to our research. And Ashok Babu is now a transplant surgeon and a faculty member, and he was able to really exploit this Donald McDonald technique of evaluating the airways. Now, why do we like tracheas? Tracheas are your windpipe, and it's not where people develop rejection, but it is an airway that has this very lacy fine network. And the nice thing is if you imagine like this Coke can, you can imagine this is your trachea, you breathe in through that way, and it comes out this way and goes to your lungs. If I had some tin snips and I was to open this up, I could turn it into a one-dimensional plane. And if you inject the animal with fitz-ulectin, which is green, prior to them dying, this lights up their whole circulatory bed beautifully. What we found to our surprise using this technique, and this is where Jingo comes in, who's in the bottom left, and I think is he here? Okay, but Jingo, you may have seen around, has been with me since I think 2008 or 2008, and he basically carried on the work of Ashok, and we had other really talented researchers as well work with us. And what we were surprised to find is that in rejection, you lose the blood supply to the airway. So in other words, when most of us who do transplant biology, we're used to just looking at a slide and seeing inflammation and saying, okay, that looks like rejection. But there's actually a physiology behind the rejection, right? And it's like, what would it be if you had rejection of the heart vessel? Well, that would be called a myocardial infarction, a heart attack. What we call these a heart attack of the airways. And you can actually use fiber optic technologies as the animal is alive, and you just put it under anesthesia. and we can put little PO2 probes and pH perfusion probes on the airway, and we can start sensing rejection. And we started to evaluate how if hypoxia inducible factor was involved with this, and we started working with Greg Semenza, who was subsequently awarded the Nobel Prize for the discovery of HIP. But we worked with him a lot during all of this work, and we remain in communication. He was really helpful. Now, if that's the case, what's the relevance to humans? Well, it turns out that the human lungs have two circulatory beds, the pulmonary vasculature and the bronchial arteries that come off of the aorta. One carries a lot of oxygen in it, and one of them doesn't. And when they do the transplant operation, they don't hook up the bronchial artery circulation. They never have routinely. And so what that means is that you're left with, I wonder if I can use an arrow. So I think I can. Oh, I can't. Okay. So you can see the red here, this is a CT scan, full frontal view of somebody who's had a lung transplant. Normally you get blood vessels coming off the aorta and the clavicular arteries, and they stop right at the place where they sew the lungs together. So we said, well, if that's the case, if we were to take a probe and touch the surface of the airways after the transplant, maybe we could detect poor oxygenation. And we did. And we had to figure out that the GI docs that who use colonoscopy, we could put one of their probes down and touch the surface of the airways of our lung transplant patients, and they were significantly hypoxic, just at baseline, even when they're healthy. So starting off, what have I told you so far? Rejection of a transplant doesn't just reject tissue, it also rejects the blood vessels, right? The microvasculature. And they already have a compromised blood circulation because they're only getting half of it. Okay, so lung transplants may not do well because their microvascular system is so poor. And Joe Su, who's now was in the lab, and this was published in STM, he showed while he was in the lab that the other consequence of that is that when you reject a blood vessel, not surprisingly, you bleed into the tissue. And it turns out that the hemoglobin, which is high in iron, deposits in the tissue and is my simplistic way of saying it, it's a beautiful food source for aspergillus. And anybody who does transplant biology knows that our patients are at risk for fungal infections, but also that the anastomosis site where the donor meets the recipient, right at that anastomosis line, is particularly vulnerable to both hypoxia, ischemia, and aspergillus invasion. So now we look at chronic rejection in lung transplantation, not just as the airways becoming fibrotic, but a heart attack of the airways. Essentially, what happens when you get a heart attack? Either you die because you get inflamed and the free wall ruptures and you bleed out, or you scar and you turn fibrotic. Well, the same thing happens in the airway, and that limits ventilation, and you don't do well. This just summarizes that idea of lung transplantation as being a guardian of tissue homeostasis in the airways, and the lung transplants may fail. We can tap into this by upregulating HIF-1 alpha, which promotes angiogenesis and new blood vessel growth. We showed in a JCI paper that that was helpful. And but we've also said the reason why you want to intervene early with transplant rejection is because once you allow the blood vessels to reject, that is actually the therapeutic point of no return. In the field, we used to say in transplant, steroid-resistant rejection. We don't use that expression as much anymore because part of the reason is, I think, because people realize it's not that the steroids it's like a special form of rejection. It's that there's something structural that has changed that make the tissue less responsive to immunotherapy. It sounds like maybe I'm mincing words, but the point is that if you don't have a good blood supply to the airways will infarct. You'll eventually grow the blood vessels back in, but it will be after the damage is done, just like an infarcted myocardium. Okay, see, I'm going pretty fast. I'm going to be done before you know it. Okay, so now I'm going to switch over to another lung vascular disease, which is pulmonary arterial hypertension. Also, it's a fatal disease without a cure that affects the small and medium arterioles. And we noticed, and I just want to attribute Norman Vocal when I was working with him in Colorado. He actually asked me, Mark, what's a good, I was working in pancreatic islet transplantations at the time, and in Ron Gill's lab. So I was kind of a basic immunologist, and he said, what model can we use to evaluate all the inflammation that we're seeing in pulmonary hypertension? And I started to rattle off some mouse models that he could use. No, they have to be in the rat. It's like, oh my gosh. So I said, well, why don't you use the athymic rat? They don't have T cells, so if inflammation is important, then they should be protected from getting pulmonary hypertension. Well, it turned out the opposite was true. They died from pulmonary hypertension much faster. And it's like, why would, if inflammation is important, you see lots of T cells and stuff, why would having no T cells be a big deal? And that's when we struck on, and of course I was in Bruce Hall's lab when they discovered the Treg. And of course I should have realized that athymic rats are predisposed to autoimmune disease because they don't regulate their immune system. Tregs are a population of CD4 positive T cells that express the genetic factor FOXP3. Sakaguchi, I have another slide I can put in here. That Sakaguchi, who won the Nobel Prize, was just down the hall from us. He was a postdoc in Gary Kaufman's lab. I had to draw a little map of him, like walking over to Bruce's lab and talking to him about this new CD4 positive, CD25 positive. This is in Grant for sport. Any of you remember Grant? And he continued to develop the field. Sakaguchi deserves the Nobel Prize. Australians were unhappy that they didn't give it to Bruce too, and they actually interviewed me for a magazine to talk about the injustice. But Sakaguchi truly deserved the Nobel for this. But back in 2000, the early 2000s, we posited that the CD4 cell that's missing in these animals was responsible. This concept was developed later, and we now know that patients with pulmonary arterial hypertension have regulatory T cell defects, and we're currently working on, I'm not sure if Dong Young is here, but he's not here. Okay, so Dong Young is working in the lab now and is showing how we can use cell therapy to help treat pulmonary arterial hypertension and correct the immune deficits. Now, okay, you might ask yourself if you're thinking, wait a second, you're using the athymic rat, there's no T cells, so what inflammation is killing the blood vessels? Why are these people having pulmonary vascular remodeling? And this is where we were so lucky to recruit from the University of Illinois, Chicago, Amy Tien. And Amy Tien, who's sitting back there, is most of you know, very important scientist for Stanford. Was an icosanoid biologist. That's what she did her PhD in. And I asked Norbert Vocal, hey, what should we study? And I got this amazing icosanoid biologist, but I don't know a thing about him. And he said you should look at 5-lipoxygenase. And so I didn't know any of this stuff. As Amy knows, I hated signaling pathways when I first started. Unfortunately, she taught me so much. And she did some really beautiful work that not only got a Science Translational Medicine paper, but also launched an international 42-center trial targeting leukotriene B4. And the way that we figured this out is that the macrophages that are well, let me a little bit about leukotriene B4. This is an icosanoid molecule that is really well known in biology as being a chemokine. I'm sorry, chemoattractant, right? So we think of it typically as a icosanoid that attracts other immune cells. It's how you get the swarming neutrophils that come into the lung during an immune response. But what Amy discovered, which is really important, is that that macrophages, when they secrete leukotriene B4, that leukotriene B4 actually kills, induces apoptosis in pulmonary arterial endothelial cells. Not only that, but it grows the smooth muscle cells, which hypertrophy, and it also grows the adventitial smooth muscle layer. So again, when you remove this, when you have this immune dysregulation, things go bad for you. But I think it really influenced the field. Amy's work did, and people are really interested now in the macrophage contribution to vascular injury. Now, what I think is our most exciting work, which we've been working on 5 years now, without it being published, but it's very close, is the work that Amy's has done looking at early B cell factor 1, which is a pioneer transcription factor. Now, for those of you like me who didn't know what pioneer transcription factors were, this is a special kind of transcription factor that opens up chromatin. It's like something like what we might have called a master regulator before. There's only about 24 pioneer factors. This one was heavily ascribed to B cells. I mean, B cell development really needed. It had not been evaluated in endothelial cells before. And we developed a novel model of pulmonary arterial hypertension and found that this new population of endothelial cells arose in the neointima, which is the thing that clogs up the arteriole and causes pulmonary hypertension. It's kind of the holy grail. What are the cells that clog up the blood vessel in PAH? If we understood that, we'd be able to treat the disease more effectively. And so because we were so surprised by EBF1, a pioneer transcription factor not associated with endothelial biology, Amy turned our lab into a developmental biology lab, which didn't know anything about, we had no experience with. And what she found was she not only characterized how the pulmonary arteries develop in embryos for the first time, showing them to be a critical factor, EBF1, but also showed exactly how EBF1 guides the rest of the vasculature. We call it kind of an organizing cell, that it guides other cells and then it dies. And then what she found afterwards is that when the cell comes back abnormally, it's normally not expressed in adults. But if you injure the blood vessel, this cell returns and it thinks that it has to basically grow a new pulmonary artery within a pre-existing artery. So this may be a fundamental new cause of PAH, maybe what's a good word, a meta cause of PAH that governs many other principles. So it's super exciting work. This kind of cartoon kind of summarizes both EBF1's role in guiding the evolution of the pulmonary arteriole, and as well as the abnormal reactivation of EBF1 endothelial cells in adults and why this may cause disease. Let me just stop for a second. It's 12:25. I guess I'm about halfway through. Go ahead.
Yes, a quick question. I may have missed it, but EBF1 shows up again in conditions where there is microvascular damage, or
So it's the link to this talk is that the cells that are affected in this disease and where EBF1 cells come from are considered the pulmonary microvasculature. The earlier part of my talk was about the airway microvasculature, but as you go out into the airways, to the gas exchanging units in the alveoli, out in the periphery, there are these cells called GCAPs, which are the kind of the universal stem cells that grow other types of endothelial cells after injury. It turns out that GCAPs give rise, we think, to EBF1 cells by the way, this is an apelin receptor positive endothelial cell, both in development and in adults. So we don't talk about GCAPs in embryology, but we do talk about them as the adult stem cell that can help repair vasculature. But in the right setting, like let's say you have a BMPR2 mutation, things go awry. And that's essentially what a lot of our papers about. Yeah.
If you don't be EBF1 expression, what endothelial cells?
EBF1 is expressed in many different cell types, but in adults, it's normally not expressed in endothelial cells. It's part of normal, it's normally expressed in endothelial cells in utero. So as a baby is developing its lungs, we believe that the EBF1 gets turned on. But the really interesting thing is that we don't know how, but we assume it's apoptosis. There's a tenfold rise in the EBF1 cells around day 14 of embryology in mice, and then they all go away. They do not become part of the vasculature. We used to call them Moses cells. We think that they help hide other cells around all over the place or organize their cells. But really interestingly, they just die. So EBF1 you find it in adults, pericytes and B cells and other, but you don't ordinarily see it in the endothelium.
So it's the basic thing, after transplantation, two different tissues, the microvasculature like from existing one in the body and one in the transplanted.
Correct.
So they meet each other like exactly like one like that, or like separately goes, where that exactly the, we talked about cyclosporin, CD4 and all this rejection.
Yeah.
How this really works actually.
Okay, so just to guide everybody back, because this is a smorgasbord of science. Right now, this is not for you, but it's for everybody else. Right now, I'm talking about pulmonary arterial hypertension. Right before this, I was talking about transplant. Okay, so your question is, we're going back to that first part of the talk, and what normally happens, like if you do a syngeneic transplant, you put a valve C orthotopic tracheal transplant into a valve C recipient. What happens is that it's really interesting. So they're disconnected, the vessels are severed, but they don't just like grow together as one tube. What happens is that you get this outstretched, like it's like that. If you held a mirror up to the Nile Basin or the Mississippi Delta, basically it's two networks joining each other like that. That's the calm, healthy response. And eventually, in a syngeneic transplant or an immunosuppressed transplant that's not rejecting, they beautifully join like two fingertips joining like this. If there's rejection that's allowed to occur, that airway will become revascularized, but interestingly, it's coming from the outside in, and it creates all of this adhesion tissue. It's sticky, it's going through fat planes. So it's actually quite hard to dissect out a rejected airway partly because of the way that gets reinvested with its microvessel supply. Yeah, sure.
So we talked about macrophages, and then those are resident macrophages, and it's also when we're talking about macrophages, triggers endothelial.
That's correct. Yeah.
Those are after the transplantation.
Okay, so this is what I was at risk for, because I'm talking about 3 different diseases today. I'm in the disease number 2. So I'm doing kind of a broad survey. So the macrophage story that I was telling you about belongs with pulmonary hypertension. And your question about resident macrophages is a very insightful one. Amy spent a lot of time on this, looking at alveolar macrophages versus interstitial macrophages. But we don't really, and we know that both populations of cells were equally capable of becoming activated and turning on. And Amy, you can correct me whenever you want.
You're right.
Okay, they both became essentially equally capable. But there's clearly many populations of macrophages in the body. I think most people consider alveolar macrophages to be resident, like you said. There's probably resident interstitial macrophages, but there are also probably monocyte macrophage populations that migrate into the lung. And it doesn't really matter, because this is not it doesn't matter in the terms of allo, right? Because this is we don't. You can correct me on this, but Fadi, but I don't. Basically, the adaptive immune response, I think, is more responsible for orchestrating the innate immune response when it comes to allo, but is that how you view it? You would hear the
It's both ways.
Okay.
So the innate response can also recognize allo and orchestrate the adaptive
Oh.
response. And there, at least in the non-lung organs, I do not know what happens in the lung. The monocyte-derived dendritic cells and macrophages seem to be important.
Okay, great. Anything? I know it's going to be really confusing because I'm going to go to lymphedema, and then I'm going to bring it all together. But go ahead.
So we are interested in the bone marrow vasculature interactions between endothelial and MSCs, which you call pericytes here. And my only paper in the pulmonary literature was with Benicio.
Okay.
On pericytes are required to interact with the vascular development. So do you think there's a role here in your pathogenesis to interact with the EDF1 positive endothelial cells?
Can I
Those
answer
pericytes?
that?
Go ahead.
So, yeah, Joy, you're right. So in fact, so Mark was just talking about like how EBF1 is reactivated in pulmonary arterial hypertension. So the cell actually retain high expression. So endothelial cell after the embryonic phase have very, very low EBF1 expression. But the cell type actually maintain high levels of EBF1 is are those stromal layer. So especially the parasite. So there are publications talking about how in bone marrow niche formation that parasitic EBF1 expression is very, very important to form those niche. And in fact, our Nature paper with reviewer number 3, I noticed that publication and keep asking me like, are you talking about the endothelial contribution of EBF1 or the parasitic contribution of EBF1?
So you're saying EBF1 is in both?
It's
population.
in both. It's in both. But what I can answer is there's definitely stromal contribution of EBF1 in pulmonary hypertension. However, when we delete in EBF1 in very particular endothelial compartment, and we see protection, and when we overexpress it in particular endothelial compartment, we basically generate PAH. So this paper is focused on endothelial cell, but there's clearly both.
I don't know if it's a challenge in the lung literature, but in the marrow, one of the issues in development is that there are some people who think there's a common progenitor for the mesenchymal and endothelial.
In the heart as well.
And so even if you are using then what you think is an endothelial specific driver, it's possible that you're hitting early on in development both?
Yeah, so the way that we did our NACA or overexpression study is we use something, instead of regular tamoxifen, we use something called 4-HT, which is a short react tamoxifen one. It's just pulse. So what we call it is it's a pulse reaction. So it only active for, I don't know, like 12 hours and then peak, it goes away. So we make sure that we activate or knock out EBF1 in adult in a very short period of time. So in development as well.
Great. Anything else? This is perfect. Okay, I'm glad to stop.
You have a question?
Yeah.
Oh, go ahead.
Do all pulmonary arterial hypertension have reactivation for the EPF1 or restricted to just one subtype?
Can you, I didn't hear the very first part of your question. Can you say that one more time?
I mean, is it reactivated in all subtypes of the pulmonary arterial hypertension or restricted to one subtype?
Oh, that's a good.
So we,
Go. This is Amy's work.
So we collaborate with someone from WashU who collect 42 PAH patients from 3 major criteria: idiopathic pulmonary hypertension, drug and toxin induced pulmonary hypertension, and
It was an shunt. It was.
yeah, shunt. I'm sorry, I'm not an MD. So the other type of pulmonary hypertension, and we are able to see activation of EDF1 in the neointimate cell across all those 3 big etiology of PAH. So I can't answer for all form of PAH or other form of PH in particular, whether that's the case. But at least in those three forms that we investigated, they have like a EBF1 activation is in the form by form.
And just to be clear, the reason why this data set from WashU was so helpful is that these are lungs from patients that have PAH. I think most of them have gone to transplantation to get, so we have their lungs, and they're specifically looking at the endothelial population, and saying, okay, what's different about these compared to a healthy individual? That's where they see the EBF1, right? Anything else? This is good.
But it's also like, so pulmonary arterial hypertension is just group 1 pulmonary hypertension. We have 4 more groups. Probably that's not studied, right? Like for EBF1, it's just the group 1 pulmonary arterial hypertension that again has like 3-4 subtypes like idiopathic, genetic, or the ones from congenital heart diseases. So they comprise of pulmonary arterial hypertension. So probably just have been studied.
Yeah.
So the thing that makes PAH different than the other forms of PH is the fact that normally when you have a lumen that's open, in PAH, you have this garbage that's clogging up the donut hole, right? And that we call that the neo-intima. That makes this particular form of PH, pulmonary hypertension, hard to treat. PH is extremely common, right? So if you have left heart failure, the pressure's back up and you have pulmonary hypertension. But it's not the way we target that is by fixing the left heart, not by opening up all the junk, getting rid of the junk. So group 1 PH is relatively rare, not super rare, but relatively rare. But we hope that this work will provide clues that extend beyond just PAH, because as a pioneer factor, if it's there in development and it's so instructive and important, if you take away the EBF1, the PAs just don't develop. But that's not the only part of the body where EBF1 may be important. And so I know Amy her future laboratory career will probably be looking beyond just the vasculature. Fair?
Yeah.
Okay. All right, so I'm going to transition now to the last disease. I think it's 1240. Okay. So which is lymphedema, which the way this happened, right? So what the heck does this have to do with the lungs? It doesn't, but we brought it back to the lungs. We had, as I mentioned, that the work that Amy did led to a clinical trial targeting leukotriene B4. And unfortunately, it was a negative trial, but it led to something that might be a positive trial here. And what happened was we've been interested in the lymphatics for a long time, largely getting back to Donald McDonald at UCSF. He was always showing that this poorly understood third circulation that accompanied the micro vessels, right? We knew that there were the standard microvasculature, but then there was parallel circulation called the lymphatics, which is responsible for immune trafficking, but it's also responsible for, it's kind of like the plumbing system for the body. It cleans up all the waste. And so we thought, well, this is highly relevant to transplant and pulmonary hypertension, but when we were getting funding, because a company kind of sprung out of this, the pulmonary hypertension work, one of the Stanford heads of Spark said, there's this guy, Stan Roxon, who I actually knew pretty well, who's got a drug that seems to work in patients, but he doesn't know why, called ketoprofen. And ketoprofen is an NSAID. And he goes, your story really reminds me a lot of his. So this is why you want to be together as large groups, because you start networking like this. So Stan had developed this mouse model of lymphedema where you cut 2 lymphatic channels on both sides of the mouse tail, and they developed something that looks very much like lymphedema. Lymphedema, for those of you who don't know, affects hundreds of millions of people around the world, and especially with people that get parasitic infections, but something like 10 to 20% or maybe more of women that get mastectomies and lymph node resections will develop lymphedema. There's no approved therapy for it. There's no drugs, and so there's really a need for it, and it's very debilitating. women that suffer this, basically have these large arms and all they can do is put compression devices on it. And there are some surgeries that are not completely standard yet, but there's no drug therapy. So trying to understand why lymphedema, another microvessel, occurs was interesting. He said, your pulmonary hypertension story really reminds us of this. And again, Amy led the charge in this again. And we were able to confirm, this is called, by the way, for people interested in industry, picket fencing, where you basically take this is the icosanoid pathway starting up with arachidonic acid at the top, ending with some of the leukotrienes down here, but also other icosanoids, and how the various drugs block things. So ketoprofen, which appeared to work as a potential treatment, the problem, Fadi may know this as a nephrologist, the problem with ketoprofen, it's got a black box FDA warning now because it's renal toxic. So you don't really, it's not an ideal cure for chronic therapy. So it really behooves anybody working in this space to try and figure out exactly what icosanoid is responsible so you can more specifically target it. And what we found was, again, just like in our PAH story, leukotriene before turned out to be the important one. We learned this just by various types of antagonists, and eventually what this is the tail thickness over time. So this was our attempt to try and do a more pictorially friendly, intuitive image where you see the skin and the tail in cross-section getting thicker and thicker over time. There's a natural kind of plateauing that occurs with this model. And then we could intervene as this was happening and see if we could make the edema any better. And it turned out if you blocked above the pathway by blocking leukotriene before biosynthesis, or if you blocked the knockout, you ended up with this great treatment for lymphedema, which would be great if it worked because it needs a treatment. Now, I'm about to show you, oh no, actually not, this won't be the next one. I'll show you what it looks like in cross-section. So if you treat, this basically shows what a control looks like. This is what a tail cross-section looks like where you haven't hurt the lymphatic channels. And then this is what saline treated control, you can see this, the space from the dermis across to the tailbone is increased. The tails get really swollen, and if you treat with a leukotriene before antagonist, you get some resolution of that. And this just shows the imaging of what this looks like. Okay, but now I'm about to show you my favorite sentimentally data that we did, at least back then before we had the EBF1 story, which is we did the necessary experiment. If this is true, then if we cut the tails, the lymphatic channels on an animal that can't synthesize leukotriene before, or it doesn't have a receptor for leukotriene before, so that's BLT1, and then the LTA4H, no, the 5LW knockout mouse, so you can't synthesize or treat. Those animals should be protected. It turned out the opposite happened. Their tails turned black and fell off. It's like, no, so it's like the worst result. Everything pointed to leukotriene before being the most important, being the most important icosanoid. We got exactly the opposite of what we expected. And so, well, just follow the data. So it's like, maybe you need leukotriene before for something. So it turned out there's this Goldilocks zone for leukotriene before. Like leukotriene before is needed, but it's needed at a certain concentration, like in the 5 to 10 nanomolar range in the tissues. And when it goes too high, it basically probably causes lymphedema. And when it goes too low, you're taking away a growth factor. And so we were able to do these in vitro assays. And also, I'm not showing you the data, but we went back and we proved it in vivo as well. So in other words, if you had a therapy that could reduce leukotriene before into this, what I would call the Goldilocks zone right in the middle here, this is a logarithmic scale here, you basically have much healthier lymphatics. So we didn't want to get rid of all LTP4 signaling. And of course, this makes sense. It's a highly conserved thing through evolution. I'm not sure how back in evolution leukotriene before goes, but it goes back a long time. I know honeybees have it. So we need this. So it would make sense that it's not all good or all bad, right? So I love that. I'm going to really breeze through this. Basically, Dong did beautiful work showing that for survival factors, lymphatic sphingosine 1-phosphate is really important. This is published in Circulation. And then Jingbo published this in JCI that HIF2 alpha, not HIF1 alpha, turns out to be a really important survival signal. I'm going fast because I want to free up time again for some conversation. Just as a side branch, we know that you need to keep the little blood vessels in your lung alive to keep the alveolar structure in place. And Jingbo, working with Shrabni, who's now a faculty member at Stanford, showed that HIF2 alpha is a really not only important survival signal for the lymphatics, but an important survival signal for the microvasculature. We originally found that when you knocked out, if you gave tamoxifen to these conditional knockouts, where you're just taking away HIF2 alpha in the endothelial cells, when we look at the airway microvasculature, it falls away. And these animals develop emphysema as well. Turns out when you smoke cigarettes, it drives down your HIF2 alpha. So this was an interesting area of research that we would love to explore further. This basically gets into the concept that I just spoke about, LTB4 being bad in high concentrations, and this talks about how you want to keep these pillars, these checkpoints in check, or in the lymphatics by preserving HIF2 alpha and S1P signaling. When you break that down, you again accentuate the possibility of lymphedema. So this is from the trial that we have. We're trying to get published now, the open label trial where Stan Squares, these patients did phenomenally well. That they're losing liters of fluid out of their lungs, their legs. And we, as we all know, we have to stay skeptical until we see the phase 2 trial results, and they're going to read that out soon. And not surprisingly, the phase 2 blinded trial, he's seeing differences, but it doesn't seem to be as dramatic as the open label trial, which just shows you the power of placebo, right? So when people believe a treatment's going to work, it usually does through mechanisms we don't understand. And this is really my last content slide, showing that what I've tried, it felt random sometimes how we proceeded from one topic to another, but I think the theme of our research since we came back to Stanford is that the microvasculature are kind of an important component of tissues that are important. It's not just static histology. Like sometimes we think of cells all existing together in this fixed plane, but actually the thing that gives vitality to an organ and a physiology to the organ. So when you actually threaten that physiology, bad things happen. That's a really simple way of saying it. So talked about three diseases today, 2 kinds of vasculature, and I just want to thank my lab again, and we can open it back up for questions, conversation.
Super interesting talk, thank you.
Sure.
I'm especially amazed by the LTB4 data, because we know in mice, platelets also secrete LTB4. Humans, I'm not sure, have you ever looked at platelets in all of these diseases? And the second part of my question would be with the Goldilocks zone. Like, it's this powerful chemoattractant, but in higher concentration, it actually leads to immune cell arrest. So could you see a difference there? Like, is it actually the immune cell infiltration driven by LTB4 that you need to see improvements or not?
Well, we have an LTB4 expert here.
LTP will work for true. Yes, you are right. Neutrophil has expressed a lot of LTB4. Macrophage, I think, also do that, and platelet for sure. But we don't do platelet.
We don't, but I'm gonna come back to a guy that who started a company, who seemed like a super awesome scientist at Imperial, and I haven't talked to him in like a decade. But he has some really interesting, the basis of the company was the bee sting. And it turns out that the bee sting has in it an LTB4 inhibitor and an anti-platelet mutant that's just preserved through evolution that allows somehow the toxin to enter but for the immune to cause immune evasion by preventing God. So there is a linkage, an interesting linkage between platelet aggregation, thrombus formation, and how I don't know why that would be packaged together in a bee sting in one molecule, but and they were trying to advocate for this molecule as a potent anti-inflammatory agent. So we can talk more about it. I'd have to go digging through again, but it was an interesting trial.
So there's the clinical association between certain lung infections and chronic rejection or loss. Do you think that's a spurious connection, not a causative one? And the cause is actually upstream in the microvasculature, like you pointed out.
Yeah, I mean, I hate to be a monomaniac where, your molecular theory, trumps everything else. Because there are clearly pro-fibrotic pathways that get activated that, we just hold this in as kind of a meta possibility. But if you think about it, a lung transplant is the biggest immunization that you could get. I mean, if you talk about a protein, nothing is bigger than a lung transplant, and so how is it that we're able even with, cyclosporin and prednisone and all of the drugs that we use, why does our body still maintain, an ability to respond to a tiny little vaccine response? There are many theories, there's a long history of large versus micro doses of antigen being involved. However, we do believe that there's a process called accommodation that occurs that somehow, I don't think it's still well understood, the body learns to, I wouldn't say ignore, because there's old literature out there to show how antigen specificity can actually promote knowledge of a transplant, awareness, the body's awareness of a transplant can sometimes help. However, we do believe, we know for a fact, like infections like CMV, and then in BK virus and kidney transplants, bring awareness, like heightened awareness again, just to use human terms, of the immune system back onto this massive foreign object. And so my guess is that what you have is accommodation and not tolerance, propay tolerance, they used to call it, remember propay tolerance? That you kind of learn to live with this thing, but then when you have an infection, it basically reactivates makes aware of what's going on. So that's my guess. And then you reject the blood vessels because of the infection.
All right, that was really fantastic, very inspiring.
Thanks.
It was exactly the right dose. Goldilocks, I'm sure. So just a reminder, July 2nd, there will be Ashwin Kumar from the Chaudhary Lab at the Amy Center. And again, August is not a vacation for this seminar. You can still get your pizza. And then September and October. So looking forward to seeing you. And again, thank you very much, Mike.
You're welcome.
It's the most people I've ever seen. You know what? Get your